Welcome back to Acing AP Biology. In this lesson, we will be talking about the chromosomal theory of inheritance. Genes are located on chromosomes in specific locations or loci. Chromosomes and genes are paired in diploid cells. During meiosis, homologous groups separate and allele pairs segregate into gametes. Chromosomes undergo independent assortment, orienting themselves independent of other homologous pairs into gametes. Fertilization restores the paired condition of both chromosomes and genes. When we previously discussed Mendel's experiments and independent assortment, we were in the context of unlinked genes, which are genes on different chromosomes. Linked genes, on the other hand, are on the same chromosome, and so they move together during meiosis and fertilization. Moreover, a dihybrid cross will not produce a 9:3:3:1 phenotypic ratio. Instead, a 1:1:1:1 ratio will arise because alleles do not move independent of each other. Genetic recombination takes place due to independent assortment and crossing over, where offspring consist of a new combination of traits that differ from parents. Offspring that are phenotypically different from the parents are called recombinants. The recombination frequency is the percent of recombinants out of the total offspring produced. For unlinked genes, the recombination frequency would be 50%. Even for genes that are linked, oftentimes recombinants do form due to crossing over, and so the phenotypic ratio may vary from 1:1:1:1. However, most of the offspring will still resemble parental types. Lastly, let's talk about map units. The probability that crossing over takes place between two genes is directly proportional to the distance between the genes. Map units measure the distance between genetic loci. One map unit is also known as a centimorgan, and it equals a 1% recombination frequency. Map units are used to distinguish the sequence of genes on the chromosome. If you increase the distance between genes, the recombination frequency increases. A linkage map shows the sequence of genes, but not the precise location of genes on the chromosome. The distance between genetic loci can be measured in nucleotides. On this chromosome, these are the different genes. Between B and BG, there are 17 map units, which means that there is a 17% recombination frequency. That's it for this video. In the next lesson, we will discuss sex chromosomes and genetic disorders. See you then. For more bio content, hit subscribe.